Uh, no, what is your presentation? I guess this one. Great. Hello, everyone. My name is Noah Greenwald. I'm a graduate student at Stanford in the Angelo and Curtis labs, and I'm really excited to tell you about the work we've been doing to develop an integrated toolkit for analyzing multiplex imaging data. So as you're all aware, it's getting easier and easier to generate the underlying data that we've been talking about. It's on the top. I know where it is. Let's just hit that new slideshow. Yeah, there we go. It's been getting easier and easier to generate multiplex imaging data. And this is really great because it means these beautiful images, which used to be, maybe a whole PhD project just to generate, there's now a variety of commercial solutions and academic solutions that can allow you to generate this data. The challenge now has moved partly, at least, from the data generation step to the data analysis step. And this is especially true with the increasing scale. We've already heard multiple talks about, the challenges of full-slide imaging, which really means that the bottleneck is increasingly becoming, what do you do with the data once you generate it, rather than how do you generate it in the first place? So I would say in order to, to go from image data to results that you can interpret, there's obviously many steps. I'm going to focus on three of them here. The first is identifying the location of each cell in your images. The second is assigning phenotypes to those cells once you've identified them. And once you've done these sort of two foundational steps, it then allows you to take this extracted data and map them to your biological variables. This is, I think, distinct from some of the other single-cell assays like single-cell RNA sequencing or flow cytometry, where the data already comes out as single cell. And so you can sort of just get started right away with your analysis in a way that for imaging data, there's these mandatory steps, really, if you want to have single-cell data, because your tissues have not actually been associated. So I'll start with identifying the locations of each cell. So you get your imaging data back. And what you'd like to know is, how many cells are there? Does the density change? All these types of questions. But what you're presented with is actually just a matrix or multiple matrices, depending on how many channels you've imaged. And what you need is something that looks like this, which is a segmentation mask. And this process of segmentation is where you identify this precise location of every cell in your image. And it's pretty easy to describe what you want to have happen, but as we've heard from all the speakers already, it's actually quite challenging in practice to get this to work. Over the last 5 years or 5 to 10 years, neural networks or deep learning or sometimes just generally AI have really made impressive leaps in their ability to generate computational solutions for what previously were much more human problems. Shown here is an example of the masked RCNN network, which is predicting a bounding box around each thing in these images, as well as coloring the pixels within that bounding box for each different object. This performance, I think, is really impressive. If you look at these images, it knows that, like, the strap of this woman's purse on her back belongs to the purse that's on her front. It's identifying the backpack that's part of the man holding the umbrella. It's getting all these different sheep. So it's really achieving really, really nice performance, and is why people have been so excited about deep learning in its potential to make what were previously really challenging problems much more tractable for analytical solutions. The caveat with deep learning is that it requires a lot of training data. And training data is where you have images of the same task that you want your algorithm to perform, and you've already done the task on those images so that there's an example of the input and what you want the output to be. And this paired data where you have images and sort of the correct answer is what these algorithms use during training in order to get this really high level performance. If you don't have this large volume of training data, then these deep learning algorithms do not work nearly as well. So this is what we set out to do in our paper, which was published in 2021. We created a massive training data set of images specifically for multiplex imaging so that you could train these deep learning algorithms. And we call this data set TissueNet, sort of as an homage to ImageNet, which is one of the foundational data sets in computer vision. What you can see here is that there was a lot more annotations in TissueNet than had previously been published because of the way we put this data set together that allowed us to really scale it to the volume of data that's necessary for deep learning algorithms. One of the crucial considerations when we were constructing TissueNet was that we wanted a diversity of different imaging platforms as well as tissue types to be present in TissueNet. This is really important because deep learning models are only as good as the data they're trained on. And so if you want an algorithm that's trained on TissueNet to generalize to different data sets, different tissue types, different platforms, diseases, you need to ensure that the data it's trained on is as representative as possible. There's no like, the programmer can't go in and make tweaks to what the algorithm is thinking. The only thing it learns is what was present in that training data set. And so having as diverse a representation as possible and making sure that your task aligns closely to at least some subset of the training data is the only way really to ensure that these models are going to generalize really well. So this is a crucial factor in constructing this data set. Once we had TissueNet, we then trained an algorithm on it to perform cell segmentation. We call this algorithm Mesmer, and I'm just going to briefly go through the workflow. You have your tissue samples, and you could have done a very highly multiplexed imaging experiment, maybe just a regular immunofluorescence. Whatever the case, you only need 2 channels of imaging data for Mesmer to run. This is really important because it means no matter what specific panel, even if you maybe had different panels in different experiments, you just need to identify what are going to be the 2 best channels to perform your segmentation. One of them is going to be a nuclear marker to identify the nucleus, and one is a membrane or cytoplasm marker to identify the shape of the cell. These images then get passed internally to the model, normalized, and the model's then going to predict for every cell and every nucleus in your image, their specific location and their boundaries. And so the result is that you take in your imaging data and you get out a segmentation mask like the one I showed before, which is going to allow you to do all this downstream analysis. So how does it work? Shown here are some example images on the top of 3 different tissue types and 3 different imaging platforms. And on the bottom is the segmentation mask from Mesmer, as well as the F1 scores. What you can see here, which I think was the whole purpose of going through this effort with TissueNet, is that the different diversity of cell types, cell shapes, cell sizes, morphologies are captured quite well with the output of Mesmer. And this doesn't require any fine-tuning in a data set specific way. So it's a single algorithm that was trained on this diversity of data that has learned something about what it means or how a cell looks, and is then able to make those predictions when you give it data. One of the really important insights that we learned from when we were developing Mesmer is that the quality of the training data is one of the main determinants of the performance of these models. So shown here is we evaluated a bunch of different models on the held out portion of the TissueNet data set that was not used for training any of them. And what you can see is that there's 3 models at the top here. So the y-axis is the F1 score and the x-axis is the speed. There's 3 models that are all sort of at this top level in terms of their accuracy. And all of these were retrained on TissueNet specifically. So once your deep learning algorithm is sort of good enough that it can learn from the data, it really doesn't matter so much what specific design choices you've made, what kind of architecture you're using. CellPose, Stardust, and Mesmer under the hood are all pretty different in terms of what approach they took. But when you train it on the same data set, they all achieve really, really good performance, basically statistically indistinguishable. Now, it's not the case for all of the models we tried. Some of them, just didn't have sufficient complexity to be able to learn from the wide range of tissue types that were present in TissueNet. But once you do have a good enough model, then it doesn't really matter which one you've picked. And the much more important criteria is what training data that model was trained on. That's really going to determine the performance. So Mesmer has been publicly available now for a year. There's a number of different ways you can access it. The easiest is just to go to deepcell.org/predict. You can upload an image and you'll get back a prediction. If you'd like, for the more computationally savvy people, you want to interact with the model a little bit more, you can look at the Jupyter notebooks that are in the Van Valen Lab GitHub DeepCell TF. This allows you to tweak the post-processing parameters. You can retrain the model if you want, get access to the weights. And if you'd like to use this as part of sort of a whole image analysis workflow, rather than just segmentation, the Angelo Lab in our ARC analysis GitHub, this is where Mesmer sort of the first of many steps for downstream processing to analyze your imaging data. Okay, so this is sort of one of the first crucial steps in image analysis. But once you've identified your cells, now comes what is, I think, maybe even a harder task, which is identifying what cell types they are. So there's a number of ways that people have been clustering single cell data. For these assays, you often have a table that looks something like this. So this could be single cell RNA sequencing data, where each row is a cell and each column is a different gene. It could be flow cytometry data, where the columns are proteins. All of these assays kind of have the same structure of this tabular data. You can take your image data, and once you've segmented it, you can turn it into a table that looks like this, because you just extract the intensity of each of your channels in each of your cells and convert it into a table where you have cells by intensity. So the question is, what is the best way to cluster it? We already heard a little bit that one option is manual gating. This is basically you're going in and you're deciding, okay, if it has this specific combination of markers, we're going to call it this cell type. You can take an approach that's more similar to what the single cell RNA sequencing community uses, where you do a UMAP, and then you kind of look at the UMAP and you circle some islands. You're like, okay, this is what the cell types are. And there's been a couple papers that have come out more recently where you can incorporate the spatial information about the cells in the image to enforce some priors on what you think the cell type is based on where it's located in the image. All of these methods, though, are premised on starting from this extracted table of information. So they all require that you input basically a big CSV file that has your cells in the rows and whatever the information is in the columns. Now, Pete already gave a great introduction to this. The issue is that defining expression of a marker by integrating the counts on the segmentation mask is often a suboptimal way of capturing what we see by eye when we look at an image. So here's an example of tryptase staining. And what you can probably tell is that, okay, this looks like a cell. There's sort of a rough halo of staining. It's a little bit stronger on one side than on the other side, but this looks like high quality signal of a cell that's going to be positive for tryptase. When you look at the segmentation mask that goes along with it, what you realize is that this mast cell is in a really densely packed area of the tissue. And so the segmentation borders are right where the membrane of that cell is. And because of this issues with the 2D section of a 3D object, there's some spillover, even with really good segmentation, where it means that when you count up the intensity of this tryptase staining of the center cell, which is the one that we think really is positive, it's not going to be so different from the neighboring cells that by eye we say, oh no, this is clearly spillover, these cells are negative. And this is the problem you're dealing with when you work starting from this, what we call a cell table, where you've extracted the counts in each cell. Your data is fundamentally not representing what by eye we see when we look at the images in terms of the distribution of the signal for each of the individual cells. You can imagine this problem gets even more complicated when you have not one, but like millions of cells across a panel of multiple different markers. You're going to have the same problem where a lot of the markers we like to use for phenotyping are on the membrane, which is the most challenging location to accurately assign to the cell they belong to. Okay, so this sounds like the perfect application for a deep learning algorithm. We have some image data with it's hard to define features. Why don't we just train a model to do this for us? Turns out that there's two problems with sort of a naive approach. One is, as I mentioned, we need a lot of training data for a deep learning algorithm. And it was hard to generate TissueNet, but it is really hard to generate manual annotations for cell clustering. It can't be the case that you get a bunch of high school students and have them circle cells, which is how we started out. You need someone who's going to have immunology knowledge. Maybe every disease, you need some expert to say, okay, here's what the cell types are. Well, this one looks dim, but really we know that often, T cells can downregulate. It's a nightmare. So it's challenging but doable to generate ground truth segmentation data. It's really challenging to generate ground truth cell clustering data of the volume that's necessary to train deep learning algorithms. The second problem is that even if we had this ground truth data, a lot of it, the question is, what are you actually going to train this deep learning model to do? It is often the case that, for different biological questions, you're going to have a different panel that you put together. And so if your deep learning model is trained to predict cell types based on a certain combination of markers, what happens when you have a different combination of markers, which is frequently the case? Are you going to have to train a separate deep learning model to predict cell types for every time you change your marker panel? What about when you don't care about T cells at all and you completely leave out the T cell markers because you have more of a myeloid focused panel? So it then becomes the case that if you're going to have to train a separate model every time you change your experiment, and that model requires ground truth data, what are you actually benefiting from? Why not just annotate it yourself and save yourself the headache? An alternative is, okay, well, that seems like a really intractable problem. Maybe we can just go broad enough that even if there's specific changes in the panel, we're still going to predict the core cell types. And the issue with this is the reason you change the panel is often because you're really interested in specific cell types. So if you go broad enough that it's going to work with any combination of markers, what's even a point of an algorithm that says this is an immune cell and doesn't give you anything more important than that? So we decided to tackle both of these problems by tweaking what the prediction problem is going to be. So rather than trying to say, have the deep learning model directly predict, here's what this cell is, we're going to do a related task, which is have the deep learning model predict, is this cell positive for this marker? So what does that look like? The inputs are a segmentation mask. It could be from Mesmer, could be from your favorite segmentation algorithm. You could do it by hand. And it's going to be the specific markers in your panel. And for one marker at a time, this algorithm, Nimbus, is going to just predict, is this cell positive for this specific marker? The benefit of this is twofold. It allows us to solve the problem of generating training data, and it means that it's generalizable to any panel that you can put together. So why does this help for training data? We have a two-tiered approach to generating training data for Nimbus. The first is what we're calling silver standard labels. So this is where we took previously published as well as our own internal data sets that had been clustered using a variety of different approaches. So, things like UMAP, Pixie, all these different algorithms people have come up with for clustering. You then take those as sort of your silver standard. And if you were just going to train a deep learning model to predict cell types, there would be no way to harmonize these different data sets because they'd have different cells, maybe the same cells defined by different markers. What we can do, though, is with biological knowledge about how those cell types were defined, we can sort of extrapolate backwards and say, if this is a T cell and this panel has CD3 and CD4 and CD8 on it, we know that this cell should be negative for CD4 and positive for CD3 and CD8. And we can do this for every cell type that was in each of these data sets and convert it into just a single channel positive or negative score for every channel, for every cell, and every data set. And this now is completely generalizable and you can have any combination of cell types in this original data that we aggregate, but now we can harmonize it all together into one really easy to train model, where this model is just learning across a diversity of cell morphologies, cell types, marker staining patterns. What does it mean for a cell to be positive, and what does it mean for a cell to be negative? Now, the caveat is that all of this training data is based on the imperfect clustering that was already done with previous pipelines. So to supplement this, we have a small fraction of this data where we have gold standard annotations, where we took the segmentation mask, we took the markers, we took a lot of grad students, and we had them just say, is this cell positive for this marker or not? And this serves as sort of a held out test set for assessing the accuracy of the model, doing the same task that the model was doing. Okay, so using this approach, we're able to generate a massive training data set of silver standard labels, almost 200 million annotations of whether a cell is positive for the markers on the panel. We were also, through the valiant effort of a lot of people looking in QPath, actually, and understand for the gold standard annotations, which cells were positive for this, for which markers, for a very, very small subset. So we can use this to evaluate how the model is doing. So how does it work? Here's an example image from a Codex colon data set where I'm just showing 3 of the markers on the panel. And then here is the predictions from Nimbus about which cells are positive for which markers. So here we've colored the predictions for each channel from 0 being black to 1 being the brightest, and we've aligned them with the same colors that the markers were using. So you can see that these epithelial cells in the middle of the image, really bright for pan-CK in the original image, and also show up super positive in the Nimbus score on the pan-CK channel. Same thing for the immune cells in CD45 and the macrophages in CD68. We can then compare that to the gold standard held out annotations about which cells should be positive for which markers. And here we've colored by F1 score. So 0 would be no agreement with the ground truth. 1 would be perfect agreement with the ground truth in white. We see that many of these cells are bright white with shades of red where the model is getting things wrong. One of the really nice things about the way we've set up this inference task is that it's not inherently learning the biases in the data sets. If you had one data set where people had systematically assigned Tregs to be CD4 cells, the model being trained on all those annotations would by definition be learning that same thing. But because we're breaking up each panel into a separate training task, the model never learns, oh, in this data set, Tregs are mislabeled. It's just learning what does it mean for it to be CD4 positive or CD8 positive or FOXP3 positive. And we see that we're actually able to get better performance from the model than the training data that it's been trained on because it is robust and generalized, and there's this really wide degree of different examples of different use cases. The output of Nimbus is a per channel score, per cell, per channel score that tells you basically what's the model's estimation of whether it's positive or negative. One of the really nice things about this score is that it displays a pretty clear bimodal distribution between positive cells and negative cells. So shown here is all of the predictions from Nimbus, and we've stratified the cells based on whether in the ground truth data, we classified that cell as positive or negative. And there's really clear separation where the majority of the positive cells in green have this nice shifted distribution to the right, and the majority of the negative cells show really clear shifted distribution to the left. This is not what the data looks like when you look simply at the extracted mean intensity per cell. And so this plot on the right, you can imagine it's much harder to understand just by looking at these values, whether a cell is positive or negative. And it's these values that are currently being used whenever people perform clustering. Whatever your favorite clustering algorithm is, or clustering algorithms that are being developed, when you're starting from the extracted counts per cell, this is what you have to work with. And so it's a much more challenging problem to even understand on a single channel level, let alone a multiplex level, what it means for a cell to be positive when your data fundamentally does not reliably communicate what we can see by eye when we look at an image. So given that, what we envision as the workflow is thanks. You can use Nimbus sort of to pre-process or transform your data and make it much, much easier to cluster with whatever your favorite clustering algorithm is. So you still get this same table where before, you have the cells in the rows and the markers in the columns, but now these values much more accurately reflect what by eye, when you look at those images, you would think for positive and negative. So here's an example of an algorithm that recently came out of our lab called Pixie. It was published this year. Grad student in our lab, Candace, did a really awesome job. We now use this to sort of take the Nimbus probabilities and perform clustering on them. It allows you to remap the clusters in a really nice way. But you could use whatever clustering algorithm you want that is now hooked up to these transformed values from Nimbus. So we're looking for beta testers. We hope to have a preprint soon coming out. It always takes longer than you think. But in the meantime, the code is available on GitHub. So if you'd like to try it out, we're definitely looking for people to break it and tell us about it, especially before we submit it. As well as this overall pipeline is going to be integrated into the Arc Analysis GitHub on the Angela Lab website. So both of the Nimbus right now just has a subclustering, but this will be in the future added in as a module to Arc, which has all of the downstream spatial analysis, as well as Mesmer currently in there. So just wanted to thank everyone who's involved with this. Geneva Miller was my co-first author from the Van Valen lab on the Mesmer effort, and Lorenz was a visiting PhD student in our lab who really led all the Nimbus work. Also like to thank the Angela Lab, Curtis Lab, funding, and I'd be happy to take any questions. Thanks. Yes.
Hey, great work. Curious on the Nimbus, how does it work on each channel separately, and whether you have multiple models with different sort of expected phenotypes that you're seeing in the different channels, or you just have one large model that kind of predicts all of them pretty consistent? Great question.
So the question was, does Nimbus take in each channel one at a time, or does it, and you have to have a separate model for separate channels, or does it, is there a model that kind of learns the interactions? We have a single model that was trained on every channel at the, sequentially. So you're going to feed it one channel at a time. It's going to make predictions just for that channel, but it's the same model that's making predictions across all of them. And then after that, you get your table where you have the predictions per cell, and you can cluster on that. Yes.
So when you trained it, what kind of augmentation did you try doing intensity modifications as part of your augmentation to make it robust against that?
Yeah, great question. So the question, when the question is what augmentations you use, and when you're training deep learning models, especially in the image analysis world, one of the easiest ways to get even more data is you tweak the images in some way when they're being trained. So you could use the same example multiple times, but it looks a little bit different, and that boosts performance. And so we basically did the whole TensorFlow like image augmentation pipeline. We did intensity, scaling, dilation, all of that is there, and that really did help make it much more robust to those types of changes. Yes.
So I really love the plots, the histogram you showed as the Nimbus score, like far outperforming the smear of regular fluorescent intensity. Have you tried like looking at that transform data with something like Flojo, where somebody could just manually like gate on cell phenotypes using Nimbus score?
So the question is, once you have this transform data, can you in fact just manually gate it? We do in the multiplex imaging that we do, it's often not practical, even if you could manually gate it to do it that way because of the, you might have your CD4 markers not quite as bright in one image. So we in general tend to do unsupervised clustering, even for like something like CyTOF where the staining really is clear. But I imagine with a lower plex panel where it would be much simpler to do something like that, it could definitely work. And the format is a CSV file, so you can import it into whatever software you want. Yes.
How does Nimbus perform with markers like PDL1? Because you have a dynamic range, you have cells that a part of the cell expresses it, the other part is not. And then a follow-up to that is you've shown us some really good examples of CD45 and pan-CK. I couldn't see a lot of macrophages in that image, but how did it do with macrophages?
So the question is, on markers that aren't as good as CD45 and pan-CK, is this actually useful? And I don't have the plots here, but we have the F1 score broken down by marker. And I would say it trends with the accuracy of the human annotation. So markers that are inherently tougher to annotate, we tend to see lower performance, but not like worse than underlying data. Something like PDL1. So interestingly, we only trained it on lineage defining markers, but it doesn't know the difference between a lineage defining marker and a functional marker or whatever. So you can put any marker in that looks at least like it's cell localized, and it will then make predictions. Yes.
So, well, I think Abhishek beat me to the macrophage question because that's always a mess to deal with. So, but one question I had was about the resolution of your data. I mean, you showed. So, how does this? I mean, has this been trained both on CyTOF and on sort of more fluorescence data set? Because CyTOF generally tends to have lower resolution. So, how did you deal with the resolution problem?
Yeah, so we did not include any like imaging mass cytometry CyTOF data in the data set for Mesmer, which we had a similar problem where different resolutions. We just have like a parameter that says like, would you like to rescale your data? It's really easy to upsample CyTOF data so that it looks more similar to 20x fluorescence. And we found that that is super robust for segmentation. We haven't tried that yet on this, but I imagine it would similarly work. Yes.
One more quick question. So in your model, you're basically getting to break it down into a binary classification scheme, but the cells are not really binary. Are you keeping track of your intermediate representation? So if it's on the last layer before you put the binary, that's a nice representation of what's going on. You keep track of that, so you could use that to do classification.
Yeah, great question. So, the classification task is binary yes or no, but cells are not binary yes or no, they're on a spectrum. Actually, what we return is the probability. So this Nimbus score goes from 0 to 1. So we are not then thresholding it to tell you if it's positive or negative. The output of the model is and the deep learning people will be mad if you call it probability because it's not technically a probability, but the values that range between 0 and 1, where 0 means no and 1 means yes, that is what we return as part of the model. So then you can decide what threshold makes sense or use it as a continuous score.
There's a question from Zoom whether Mesmer was trained on 2D or 3D images.
Mesmer was only trained on 2D images. And we have not, the follow-up question is, what do you think about 3D? I think 3D is really hard. And so far what we've found is that if you want to have a whole PhD project on a 3D model, That's option one. Option two is just take your 2D slices and like stitch them together, and probably that'll be good enough. And at least it's gonna be many years less work to do. So I would definitely encourage people to try that one first, and only if it doesn't work, think about doing the 3D model. Right now, the Mesmer model will not learn based on one stack, change its prediction on another stack. But if you have good enough performance and you're okay with some error rate, then that seems to work pretty well. Yes.
I'm curious as to how the Nimbus deep learning model actually works. Do you predict sort of like a dense prediction across the whole image per pixel, like this like probability of being positive for the market per pixel, and then you average the cell? Or do you take the cell and input to the neural